Okay, we're now going to continue with our examination of this whole fluid mosaic model. The last lecture, we discussed the mosaic portion of it, the integral and peripheral glycolipids and glycoproteins that may be present in and on the membrane. Well, in this lecture, we're going to be looking at what it takes to maintain that fluidity. Okay, the membrane has to stay fluid so that things can pass through and things can be maintained inside, outside the cell. We see that fluidity has three primary things that influence it. Three. One, phospholipid types. Okay. It's not so much what was the R group on top of the phosphate on the head of the phospholipid, but it's going to be, well, was it both fatty acids being saturated, or was one of them unsaturated? Depending upon the ratio of saturated versus unsaturated, the membrane can have more or less movement in it. And that's the thing. We want the individual phospholipids to be able to move, spin in place, and move left and right. Because as they're able to move around, so are the glycolipids and glycoproteins. They can come together and they can disperse out across the surface. This fluidity has to be maintained for each type of cell, for each species of animal, for each different species of organism. We find that the levels of fatty acids that are saturated versus unsaturated, the more saturated they are, means the more straight, that means the more the phospholipids are packed in together. There's less movement, less spinning. If they're all like this, nothing can fit through. Nothing can be inserted. Metabolism's going to shut down. And if they all have at least one of the fatty acids bent, well, now there's too much space. They're too wiggly. Things move too much. Cannot maintain an inside-outside. Stuff leaks in, stuff leaks out. Cannot maintain a metabolism that way. So you have to have a good ratio. The ratio is going to be dependent upon number 2, temperature. Over millions of years, we mammals, well, actually pretty much all animals to a certain extent, have evolved to have a certain temperature. Us, 98.6 degrees Fahrenheit, 37 degrees Celsius. That's our internal temperature. Other mammals, other animals are going to have different. You have to remember what we call temperature is nothing more than a way to gauge, to describe and discuss how much thermal energy is or is not present in the environment, whether that's the environment outside or internal. 37 degrees Celsius, that's what we need to maintain. It's a Goldilocks thing. Too hot, too cold, just right. If our internal temperature gets too hot, what happens is no matter what the ratio of saturated, unsaturated is, so too much thermal energy means the molecules are going to be moving. The phospholipids are going to be moving too quickly, too fast. They're going to become leaky. Stuff's going to leak in, stuff's going to leak out. Your cells are going to die. Cells die, tissues die. You die. Too hot, too cold. You start suffering from hyperthermia. Your temperature's dropping. You get too cold. Hypothermia sets in. Your internal temperature is dropping. 35, 34, 33. You're now getting close to dying. Why? The lack of internal thermal energy means that those phospholipids are now slowing down. They may have space, but they're too slow. Things that should be able to cross across the membranes can't. Cellular metabolism begins to shut down. The just right of our too hot, too cold just right, 37 degrees Celsius, plus or minus a degree, depending on the person. Our membranes work the best at that temperature. And to help maintain that temperature, cholesterol, or I'm sorry, not the temperature, that fluidity state. Cholesterol. Yes, I know you've heard cholesterol bad. Well, no, you need cholesterol. We make pretty much what we need, but if you're eating other mammals, you're going to be getting cholesterol. Cholesterol acts as a spacer. It pushes and helps maintain a specific distance between phospholipids. Not all phospholipids, it just inserts throughout the membrane randomly here and there, helps to maintain, give a little bit more space. Now when you look at the plasma membrane, you're going to see that it's, asymmetric. It's not mirror images on both sides. If you were to go through and if you were to draw a line between the phospholipid tails, you get an outer side and an inner side. Outside, inside. Some textbooks still refer to this as an outer leaf, inner leaf. It's kind of falling away, but it helps describe what's going on here. The way they used to study, and the way they still, to an extent, study membranes, is they would freeze cells. They would freeze the cells, and they would take a diamond-edged knife, or diamond knife, and they would peel it back. Peel back the two parts of the membrane. When they were doing this, it reminded them of opening a book. Pages in a book used to be referred to as leaves. Outer leaf, inner leaf. Outer leaf, inner leaf. Outer layer, inner layer of the lipid bilayer are completely different. You're going to see differences in how much cholesterol may or may not be there in the two sections. You may see that the glycolipids, glycoproteins that are associating with outer and inner is going to be completely different. You're all definitely going to see that the peripheral proteins associate primarily with the inner membrane, the inner half. The cytoskeleton is going to be attaching to peripheral membrane proteins, helping to maintain the shape and the size of the cell. Now, the cell has to be able to get things inside, outside, outside, inside. We call this transport. When I say the word transport, a lot of people seem to think of moving stuff around with a truck. You're transporting it. You're moving it from point A to point B. Just turns out that point A is one side of a membrane, point B is on the other. Have to keep in mind, the membranes themselves don't stop everything. They do allow some things to pass through. Because of this, we refer to them as being selectively permeable. Permeable means it can pass through, allowing things inside and out. Selectively permeable, well, only certain things can. Some of the things that we find that can pass through: water, gases, oxygen, carbon dioxide. We're going to see also, and we're going to discuss that the selectively permeability means that larger molecules, sugars, proteins, cannot pass through. The selective permeability means that nonpolar, water-hating, can pass through. Because think about it, where, like I said in a previous lecture, there's no water there. So nonpolar molecules are going to want to go where there's no water. Polar molecules that love water are not going to want to leave water to go into an area that has no water. To get things to move across the membrane, you either have to do work or you have to let the state of matter do the moving for you. One of these we refer to as passive transport. No energy, no work has to be done. Whereas active transport, well, work has to be done. ATP has to be used. When it comes to passive transport, we're letting the natural state of matter move things across the membrane. Diffusion is the state of matter wanting to go from a high concentration to a low concentration. No work needs to be done, it just happens. High concentration in one place, low concentration in other. The molecules here have a higher number per set volume than over here. Given time, these molecules will move all throughout the area, filling up the volume and equally dispersing. Great example of this. You walk into a room, you catch a whiff of something. You're not quite sure what it is. You start smelling around for it. You've done this. You move one part of the room, don't smell it over here. You move to another, smell a little bit more here, and you keep moving around. Keep moving to where you smell it more and more until you suddenly find whatever it is. How is it that those odor molecules, those scent molecules, are more pronounced in one part of the room than the other? Well, diffusion. Around the object of whatever it is that's giving off those odor molecules, in its close vicinity, you have the higher concentration. The further away you get, the lower the concentration. So what we see is, given time, those odor molecules slowly move throughout the room. It's random which direction they're going to go. They're just going to start filling in the room. And before you know it, wherever you are in the room, you smell it, and you smell it equally. Well, as you see here from this figure, membranes utilize the same thing. Whatever those bluish green things are supposed to represent, whatever molecules they are. You see on one side of the lipid bilayer, the membrane, they're at a higher number than on the other side. Given time, those molecules will slowly diffuse across. We start to see one side of the membrane, the other. As the molecules move, it starts to get more and more equal until we hit a certain point that they are completely equal. For every one molecule that moves this way, another molecule moves that way. We have now reached what is referred to as equilibrium. Equal number on one side or the other. This is passive transport. Notice, no work has to be done here. No ATP expended. Downside of this, once the equilibrium gets maintained, well, for every one goes this way, one goes that way. You'll never get an unequal division again. Now, things that drive this diffusion can be concentration. You know, if one side is only slightly higher than the other, well, it's going to take a lot of time for them to get. But if it's really way off, we're going to see that diffusion is going to occur at a faster pace, a more accelerated pace. Mass of the molecules, it's just one of those things. Smaller things move faster than bigger things. Temperature, how quickly are those molecules moving? If they're moving faster and faster because, you know, there's more thermal energy. Not only are the molecules that want to move across the membrane moving faster, because remember, everything is vibrating, Brownian movement. More energy there from thermal energy means they're going to be moving faster and faster. They're going to diffuse quicker. But also means that the phospholipids are moving faster. They're getting leakier. Things can move across faster. If it's cold, well, less movement all around. Things diffuse slower. Then you have to take into account solubility. Polar versus nonpolar. How much distance has to be traveled for this diffusion act to occur? You know, we see that many of our organs, our kidneys, you know, it's a great example, use pressure to force things across membranes.